And I definitely want to get into the different assays in a minute, but first I want to tackle a few process topics, namely, with changes to the process, how do you prevent that from delaying production if you're moving and shaking and things are changing as you go? Deepa, do you want to address that first?
I think from the quality perspective, I would say keep a good change control process in place. So when you're making changes, you have a robust system to track the changes down. So when you see differences in your product or you differences in the characteristic, you can point it out to those changes versus just blindly keep moving and keep changing. That would be, I think my perspective. I'll let Doug and Hoyoung talk about this.
Yeah, I think this is an interesting, this is probably one of the biggest challenges that I see from our tech ops group is to say, what methodology should we use? Because we want reliability, we want reproducibility, we want the scale to be appropriate for the demand. And that almost, at least today, in many cases, it almost steers you right towards the tried and true solid phase peptide synthesis. And so if you'd use that approach, you know that it works. People have used it for many years. And the big challenge then is just, can you scale it to the appropriate level? My head of drug substance tells me all the time, in his early career, a big campaign was, 10 grams of a peptide. And now we're making, 10 kilograms, 20 kilograms, 50 kilograms at a time. So there have been process changes that have been introduced into SPPS, but in many cases, those are much more well developed and mature than some of the other approaches that we talked about earlier. So I think if we decide you want to make a change away from SPPS into LPPS or fragmentation or even recombinant, it is, to Deepa's point, very, very important that we confirm that through the change control process that we have absolute oversight over everything that's taken place. And then importantly also that we have confidence that that change is going to make a realistic difference, because the last thing you want to do is sort of fix something that isn't broken, but you also want to try and advance and continuously improve. So it really is, it's a tricky balance besides, when do I want to change to one of these other technologies and approaches? And that becomes not just a technical decision, but also a business decision from a risk standpoint, because you better be doggone sure that you're going to have this new change or this new process improvement work before you make it, because if you don't, you risk being stocked out of your product, that's for sure.
Definitely. And Deepa, you had mentioned earlier, sorry, did you?
Yeah, I was just going to add to that, just making these changes in the process. The other important thing is to make sure you have good methods in place. So when you're making these changes, you can truly evaluate the impact of this change. So I think that's why it kind of brings it back. We need to spend some time developing the method appropriately, which is giving us the right value or right characteristic of the molecule we are looking for. So when you make the changes, you can easily differentiate, did it make any difference or not? Go ahead, Kate. Sorry, I stopped you in between.
Oh, I just wanted to jump in a little bit to batch versus continuous, because you had mentioned that earlier. And as we talk about process, what are the trade-offs between batch and continuous manufacturing for GLP-1 analogs?
You have to look at from both pros and cons point of view. At the early stage, when you're developing your molecule, you're in the early stage of development, I think batch processes are the best way to go. But once you have full-fledged developed process in place, continuous manufacturing is a great way. Obviously, it comes with a lot of risk. You have to monitor your process as you go along. It definitely speeds up your process. But on the other hand, you're taking the risk of losing the batch. If something goes wrong, then you end up losing the whole batch. Versus when you're making small batches, then you're only limiting to that small batch. If there are any issues, if there's investigation needs to be taken care of, you're holding the one small batch versus the continuous manufacturing process. When your whole batches, which is much bigger in size and much more in value, is on risk. So I think it goes with, what stage of the development you are and how much risk are you willing to take. But FDA supports now continuous manufacturing, so which is definitely a great thing especially for the established organization and the established processes.
I think I think that response is spot on. I will say that we are just now investigating approaches that we could use continuous manufacturing in all or part of our products. I think because we have a small molecule oral, we might be able to have a more applicable approach using continuous manufacturing and that than we do for the peptides. But we're certainly evaluating it on both sides because, as you said, there are pros and cons to both sides of it, and you just have to be sure that your particular process is amenable to a continuous manufacturing approach.
And I know, Doug and Hyung, I know you're at different parts in scale up, but I'm interested to hear, what sort of bottlenecks have you seen so far, as you approach scale up?
So I will say that early on, and it's to Hyung's point earlier on, the lead times on certain things just even a year ago were frightening. I'll never forget the first time we went to the CDMO and said we need to get syringes for a pre-filled syringe, and they said the lead time was 62 weeks. I said 62 weeks. That's not, I can't make that
it's
work.
Over a year.
Yeah, exactly. And that was only to get a partial delivery of an overall order that we wanted to put in. So I think from that standpoint, things have gotten a whole lot better. So we're not seeing tremendous, or at least something that's not manageable in terms of lead times for stuff. I will say that if you have to do scale up on equipment, though, if you need to add change parts or something like that, there still seems to be quite a long lead time on those things. So that's a hurdle that we have towards scale up, is to making sure that the process train or the manufacturing train itself is set up to produce at the scale that we want to. But the biggest thing that we have is just, like I said, getting all the components that we need and then turning the crank, because when you're using solid phase peptide synthesis, or if you're in batch mode or whatever you're doing, if you can't continue to keep that crank turning, then you're going to be in trouble. So one way to combat that, of course, is to have multiple suppliers, to have a network of different manufacturers who can each contribute to the overall need that you have for scale up. But I'll tell you, Katie, it's not trivial right now. To manufacture these quantities of peptides that we need, you really have to be thoughtful, and you have to have very reliable CDMO partnerships, and you have to emphasize to them that this doesn't mean that CDMO A is better than CDMO B or better than CDMO C. It's just that we need all of you A, B, and C to deliver at the same time, so that we have that continuous supply that we need at the scale, until someday when I can afford to build a $3 billion manufacturing plant of our own, which is not going to be next week, that's for sure.
Very good point, actually. I think the other bigger issue is making sure that you have contract with CDMOs in place, because depending upon when you reach out to them, they put you in the waiting list, and then you're waiting on their mercy. How do I really coordinate between 3 different CDMOs to make sure my material is made? I think another thing I was going to add to that is another bottleneck which I've seen is testing. So testing sites, if you have a different CRO who is taking care of the testing, you're dependent on them. Like where do you stand in their queue, right? Are they going to be able to do the work quickly or not, unless you bring in the expertise in-house? So, something to think about. How do you want to really manage that scenario?
Okay. From us, it was in the early stages, it did take some time, as Doug mentioned, the purities, impurities, catching those, updating it, and trying to match the regulations. And those were the hardships. And I think, as Deepa said, it's very important to set up those processes in the early start rather than in the later, because as it goes, it can impact your schedule with the clinical trials.